Hello everyone, my name is Dyandra and this is my research partner Amrita. We would like to present to you our findings regarding our discovery called AntiNeptic, where we created an antibacterial wound spray through the enhancement of Nepenthesin enzyme from the genus Nepenthes under different concentrations of calcium chloride.
Nepenthes is known as one of the most unique plants that can only be found in tropical rainforests, one of its densest populations residing in Indonesia. Their resilience to nutrient-poor environments calls for a unique mechanic of pitfall trapping insects as a source of nitrogen. In its freshly opened state, its digestive fluid possesses a rich composition of enzymes and antibacterial properties, including an aspartic protease known native only to the genus Nepenthesin. In general, fluids from unopened pitcher plants consist of 25 millimolar potassium chloride solution, enzymes, defensive proteins. These compounds of the digestive fluid indicate the plant's ability to react towards microbes. Proteolytic content can be partially isolated by measuring activity through exposing specific substrates to Nepenthesin enzyme, such as casein through casinolytic assay.
Our experiment involves assessing the enzyme qualitatively and quantitatively. Qualitatively, we try to observe clear zone diameter through agar well diffusion. Quantitatively, we assess the Nepenthesin enzyme through absorbency. Concentrations of calcium chloride will be manipulated to serve as a catalyst in enhancing the medicinal properties of the aspartic protease Nepenthesin. Finally, our final product will be in the form of an organic antibacterial wound spray. Here are our two main issues that turn botany into benefit: dangerous chemicals and Indonesia's diversity.
Our hypothesis states the higher the concentration of calcium chloride with an optimum performance of Nepenthesin at 8 millimolar, the higher the absorbency reading, which indicates higher tyrosine presence and wider diameters of clear zone. To support this, we formulated an experiment consisting of 4 main phases. Our pre-lab contains 3 subphases such as agar preparation, sterilization, and standard curve preparation. This process is vital to seeing the macro visualization of the Nepenthesin proteolytic activity, and is important that lab environment is strictly kept to avoid contaminants. In this phase, agar solution was prepared and poured into Petri dishes. 5 wells were made with labels. Nepenthesin enzyme and calcium chloride variables were inserted to wells along with 2 types of controlled positives of industrial proteases and pepsin and a control negative filled with distilled water. The hardened and filled wells are then left to sit for 24 hours.
The third phase is the casinolytic assay phase to gather data quantitatively through absorbency and using the spectrophotometer at 660 nanometer wavelength. To prepare the solutions for the assay, a series of incubation periods and chronological input of multiple reagents was crucial to the completion of casein and Nepenthesin reactions. Solutions were put into cuvettes and tested for absorbency. Here is the image of the free tyrosine standard curve produced from tyrosine standard stock solutions. According to Beer's law, absorbency is directly proportional to concentration, and standard curve is used to measure unknown concentrations of unknown solutions. The last phase is combining the most optimum concentration of calcium chloride with constituents like neem oil, witch hazel, and Nepenthesin together into a spray bottle. Here are the results of the experiment.
The first 4 concentrations of calcium chloride, 1 millimolar, 2 millimolar, 4 millimolar, and 6 millimolar, shows very faint halos or clear zones surrounding test holes 1 and 2, which contain a Nepenthesin enzyme with and without calcium chloride, but are apparent upon close inspection. However, there are no incremental changes as the concentration of calcium chloride increases. The left shows the skimmed milk substrate of the 4 millimolar variable with a visible clear zone on hole number 2, which is Nepenthesin and 4 millimolar of calcium chloride. As seen, the clear zone is comparatively similar to that of the industrial pepsin, which is a controlled positive enzyme. This indicates that there is potential in the 4 millimolar variable, however, only occurred in said trial. The right shows the 8 millimolar calcium chloride variable, which has a wider and a more vivid clear zone compared to the preceding concentrations. Qualitative data is meant to provide a macro visualization of enzymatic activity that may be used to assess and pre-indicate possible enzyme activity, such as that belonging to the Nepenthesin enzyme, and property judging by substrate compatibility.
Here are the table of absorbencies. Before organizing data into graph and to see its trend, data was translated into tyrosine concentration using the equation from the standard curve, such as the following presented on the screen. After translation of the average of absorbencies from each variable, we acquired the following data. Data suggests that there were incremental changes where higher tyrosine concentrations from higher absorbency readings increases as calcium chloride concentration increases, reaching 8 millimolars, which is the most maximized. The R-square value is shown to be 0.7946, which is a good correlation. It is also important to note how the wide spectrum of areas of pitcher plants may affect the pattern of the graph. Here are some visuals from what the sample looked like visually. Notice that in the far right picture of the 8 millimolar variable, it shows a deep blue color, which indicates good enzymatic activity and high amounts of free tyrosine after casein breakdown.
Analysis of variance is a way to determine whether there is correlation between the means of 3 or more independent groups or variables. The H0 or null hypothesis suggests that there is no difference between the means, while H1 suggests that there is difference between the means. It is important to note that H1 can only be accepted when the A, which is the probability of rejecting H0, A is less than 0.05, meaning that there is correlation between independent and dependent variable. If A is greater than 0.05, no differences are found between variables. As seen in the table on display, the p-value is less than 0.05, meaning H1 can be accepted and H0 is rejected. The value between F and F crit can also be considered, where there is a difference in value signifying validity of data.
Validity is affirmed by the ANOVA test. Additionally, the test of homogeneity of variances may be conducted. Similar to the rule of p-values, the significance value has to be below 0.05 alpha in order for the H0 to be rejected and to accept H1, where H0 denotes that there is homogeneity in data or no difference between means, and H1 denotes that there is difference within and between means, and that data is not homogeneous. When data passes the test of variance homogeneity, the final test, called post hoc, can be conducted to compare the differences between multiple means from each other. When there are significant differences, it is marked with a star sign. The more stars are found, the more significant the concentration of calcium chloride are in effect towards Nepenthesin activity.
Given the data results mentioned previously, the 8 millimolar variable will still be the concentration of calcium chloride utilized as the catalyst ingredient within the final product, given that Nepenthesin is best maximized with 8 millimolar of calcium chloride, producing the highest amount of free tyrosine. The final product will be in the form of an organic wound spray.
The organic wound spray is comprised of neem oil, witch hazel, pure water, 8 millimolar variable calcium chloride, and finally the Nepenthesin fluid as constituent. The end product forms a milky solution with a prominent smell of neem oil, which resembles the smell of shallots. The spray was tried on the skin of the researcher, and no irritation was felt after a period of more than 10 minutes. In closing, data results conform to the hypothesis where the higher concentration, nearing 8 millimolar calcium chloride, the more optimized Nepenthesin enzymatic activity will be. Graph trend shows inclining and positive correlation and conforms to past research papers that have investigated the relationship between divalent metal ions and proteolytic activity by Chitur et al., showing similar trends that holds true for the Nepenthesin enzyme as well. Unfortunately, we were not able to carry out any bacteria or minimum inhibitory concentration experiments due to limitation of the pandemic. This had limited the scope of the experiment. However, we could guarantee that the strength of our experiment lies in the sterility control as well as discipline to the methodology, therefore increasing the likelihood of producing anticipated results.
The organic spray wound remains a prototype, and no wounds were exposed to the spray in order for the experiment to remain ethical and to avoid infections due to chances of impure substances. In future works, it is hoped that the organic wound spray can be processed better in pure state. The experiment could also be expanded by extensively purifying the pitcher fluid and isolating the Nepenthesin enzyme for more controlled reactions. Various substrates could also be experimented on to uncover the more enzymes or expanding on multiple methodological approaches like chromatography and circular dichroism. Finally, multiple species of Nepenthes may be explored to see the various factors such as viscosity, age, enzyme concentration, and chemical constituent.
Here are our references. We hope you enjoyed our presentation video. Thank you for your attention.